Okay, so this is chapter 1, which is actually talking about cell structure and function. So this is going to be more normal kinds of function that you would expect to see. It's supposed to be a review, so you can kind of know what a cell is supposed to do. So then when we start talking more about how the cells are misfunctioning, that a lot of this will make sense. So hopefully this is a review for a lot of you, if not most of you. Okay, so this word right here is very important, homeostasis. Homeostasis means that the cell is trying to maintain a predetermined uh, level, like your body temperature, for instance, is supposed to be 37 degrees or 98.6. So 37 degrees Celsius, 98.6 Fahrenheit. So that's supposed to be the normal body temperature. That doesn't mean that um, some people don't have this being slightly off, and that's their normal. So that's maintaining homeostasis for them. If their body temperature is normally 99 Fahrenheit, then that's their normal. Salt and ion concentration. So where we get into the difference is if we have a cell here, sodium is at a very high concentration on the outside of a cell, and that's true for just about every cell. There's an exception in the ear, but for the most part, every cell has a high concentration of sodium on the outside and potassium on the inside. Okay, and that's homeostasis. That's when things are normal. Okay, they're not at equilibrium, so that means that the sodium isn't the same concentration inside and out, and same for the potassium, but they are at homeostasis. Okay, so those are salts, and chloride's the same thing. Um, it's pretty much higher on the outside than it is on the inside. Okay, so that's what we mean by salts. Salts break apart to form ions. Osmotic potential. Now, osmotic potential is a little bit different because water, if these little dots are water, cells are always at osmotic equilibrium, and that is their homeostasis. Okay, so they're always at equilibrium because the water is able to move in. So if we have an addition, and we'll talk about this again later, of particles inside the cell, okay, so the little green dots are supposed to be particles that can't penetrate, okay, proteins, DNA, all of those things that are inside a cell, um, then what will happen is water will tend to move in to try to offset that concentration because water is everywhere. And that would then cause that cell to expand, so the concentration would be the same inside and outside. Okay, so since the particles can't move, then the water would then move in, and that would cause the cell to bulge out. Okay. All right. So osmotic potential, yes, you are always at osmotic equilibrium. Okay, and that is homeostasis. That's where it's supposed to be. pH, everything likes to operate at a certain pH. We'll talk more about that. And by everything, I mean enzymes, protein enzymes, and channels, and that kind of thing. DNA, mRNA, ribosomes, all that stuff likes to work at a certain pH. Okay, um, so you have to maintain these guys here, but you also have to have the right molecules. Nutrients is what we would call those. We have to have the right nutrients. We have to have a good supply of glucose. Okay, so glucose has to be able to move into the cell, and you have to have a constant supply of that. Minerals, now when we talk about minerals, we're talking about mostly our ions: sodium, potassium, chloride, magnesium, iron. Those are all minerals. Vitamins work as cofactors or coenzymes for enzymes that do things. So we have to have a steady supply of these, and we have to have amino acids because we have to make proteins, and proteins are just strings of amino acids. Okay, so we have to have a constant supply of amino acids. Okay, so that's to make a cell function properly. Wastes have to be broken down. Okay, so you may increase your amount of a certain enzyme because of something that you're doing. And so your body may say, oh, I need more of this enzyme. And then afterwards, when you stop doing whatever it is you're doing, whether it's running or whether it's, I don't know, whether it's something that you've eaten or drinking, that's a big one, especially alcoholic beverages, increase the production of enzymes, and then after a while you have to get rid of those. So the question is, what happens to those? Well, they have to be broken down by the cell. So that means that you need a cellular digestive system, which is where we'll talk about lysosomes, proteasomes, and peroxisomes, things like that. Okay, so when these are all in the proper range and proteins and membrane components are being made properly, so if everything is working great, then the cell will remain in homeostasis. Okay, so then you're maintaining homeostasis. Now, what we're interested in is this for this particular class. What if things stop working properly? Okay, when any of these things are not working properly, tissue and organ systems may fail, and that's what we call pathology. So that's what we're studying. We're studying the physiology, but physiology, what you've learned so far, has been about things working properly, and now we want to say, okay, this is the way it works, and that's what we're going to spend this video doing. And then in the future, we'll start talking about, okay, well, this is what's going wrong with things. Let's see. Pathogens, mutated genes, environmental influence, etc., can interfere with cell function. So we're going to introduce some of those things. Okay, so this is a typical cell. It has things like cilia sticking out. Cilia are the things that whip back and forth to move fluids. Microvilli, which are increasing surface area. The mitochondria, which is making ATP, specifically in the presence of oxygen. Okay. Ribosomes, which are going to translate proteins. Peroxisomes, which I mentioned are the peroxisomes, lysosomes. These are part of the cell's digestive system. And then microtubules, which are giving it structure and shape. Okay. Endoplasmic reticulum, there's the rough endoplasmic reticulum, the smooth endoplasmic reticulum. The rough endoplasmic reticulum is where proteins are being made, specifically proteins that will be excreted, like these guys up here. Okay, so they'll move through the endoplasmic reticulum and then through the Golgi apparatus, and then they'll be excreted. Okay. All right, so that also mentions the Golgi apparatus. Okay, the Golgi apparatus is where these proteins get cut up and activated, put in their active form, and then they can be excreted, or they can be joined up with something like the peroxisome. We may need to make an enzyme that the peroxisome is going to use to break things down, so it'll go through that process and then go to the peroxisome. Okay, and then the cell membrane, which is surrounding it all, and of course we can't forget about the nucleus. Okay, the nucleus that sits in the middle. Okay, so I think I hit on most of these things. Secretory vesicles. Vesicles are just those are the things that are holding those guys there. Nuclear pores. That's how something called mRNA gets out. All right, so let's look at some specifics. The membrane is mostly made of lipids, with proteins scattered throughout. Now, when we think about what's surrounding a cell, usually, I know I do, I just draw it as a circle. However, it doesn't really look that way. It's got lots of proteins in it, okay, lots of different proteins, different types of proteins. Some are just stuck to the outside, some are going all the way through. And it has cholesterol in it. So cholesterol is another thing that you kind of find sitting down inside the membrane, and I guess I should stop drawing that because there's a nice picture right there. So you can see the cholesterol here that's scattered around. You can see here's a pore that things can move through. So if you want to let ions in or something like that, then you can open up a pore. Aquaporins let water through, so you can have things move through that pore. Okay. Sometimes it's only in one direction because of concentration gradients, but certain things, and it can be selective, can move through this. Here's a peripheral protein that could be, or here's a peripheral protein on the periphery, and this is an integral protein that's moving through. Okay, so lots of different proteins, and these guys are all doing something. They're not just there to make it look nice. This might be, in fact, this guy right down here, this peripheral protein might be an enzyme, and it might be breaking things down. Okay, like cyclic AMP is one example of that, if you've ever heard of that. Okay, and so we also see that we have something called glycoproteins. And glycoproteins are pretty important because that's one of the reasons we want to control our blood sugar. That comes back to homeostasis. That's one of those other things that we want to keep. We want to keep our blood sugar in a nice tight range. We don't want it fluctuating wildly. We want it at a constant level, because what'll happen, and I'll make this green, is if you have too much sugar, you might have a buildup of sugars on these proteins or on these phospholipids or uh, glycoproteins, glyco meaning sugars, and glycolipids. Uh, so you might have a buildup of sugars on those, and that's, I think I mentioned in class the A1C test, where they actually test to see how much sugar, how much of these glucose molecules have built up, to figure out if your blood sugar has been controlled or if you've been cheating, if you're diabetic. Um, they'll actually test this to see how much of these sugars are building up. And it doesn't necessarily mean you've been cheating. I don't mean to imply that. It may mean that your diabetes is very difficult to control or that your medication isn't working properly. Okay, so this is another picture of the cell. It's just an alternate picture. It shows again the endoplasmic reticulum, rough and smooth, um, and then the Golgi, and then what's being secreted here, and then your mitochondria that are sitting here, and then your digestive system, your lysosomes and peroxisomes that are in there. Okay, so I think the easiest way to go about understanding what's going on in a cell is to actually make a protein, so to speak. Okay, so proteins are, I often like to ask the question, um, does lettuce have proteins? Yeah, because people tend to say, well, lettuce is not a good dietary source of proteins. If I need a lot of protein, I'm going to, I'm going to eat meat or something like that. Um, but remember this, DNA, the whole job of DNA is to code for proteins. So if something has DNA, which all living things do, DNA or RNA even, all living things have DNA, then it's making proteins, because proteins are the things that that perform all the functions inside the cell. They're the things that make these things that are being secreted. Insulin is a protein, for instance, and this is a picture of insulin. And all it is a string of amino acids. Okay, so we know that we need amino acids, and we get most of those through our diet, but we do have protein enzymes that can convert these from one to the other. In many cases, I don't want to point at that because I don't know if threonine gets converted into lysine or not, but we do have enzymes that we can make, and we have enzymes that we have to get from our diet, essential or non-essential, is what those are, if you recall, or if you've ever taken nutrition. Okay, so what we're trying to do is put these guys together in a certain order. Okay, and that's where we kind of get different. So in certain places that are very important, you have to have the amino acids exactly in the right order. But some of us have a little bit different sequence. I don't know how much that would change for something like insulin, but we might have a different DNA sequence that gives us, maybe instead of a glycine here, maybe we get an arginine or something like that. And if it doesn't change the structure, then we're okay. If it does change the structure, then that may not be something you can survive. Okay, so that's a deleterious mutation in the DNA that's coding for this amino acid. Okay, so remember that the DNA is coding for the proteins, and the protein is a string of amino acids. Okay, so the DNA is coding to give you that sequence of amino acids that it has to line up in. Okay, all right, so shape matters. I have that written here. A lot of times it's the shape of the protein that gives it a majority of its function. Now, you also have to have certain amino acids that have different chemical properties, but in general, the shape has to be maintained for it to function properly. The other thing is that temperature, pH, toxins can also interfere with the function of the protein. This protein right here was made to work at about a pH of 7.4. Okay, so if your pH gets out of range, if it goes too high, alkalosis, or if it goes too low, acidosis, then not just your insulin. I don't even know how your insulin would react to different pHs. It probably wouldn't like it. But there are many proteins, and inside the cell, especially if you have a buildup of lactic acid, which we'll talk a little bit about, then suddenly these proteins will not function right, and that can cause more effects, and it can almost be kind of a snowball effect. So we want to maintain our pH for that reason too. Okay, so if we quickly go through this concept of, or actually it's not a concept, it's something you do all the time and you're doing right now. But if we think about what goes into making a protein, remember the code exists here, and this is the DNA. Okay, so this is your DNA. It's all unwound most of the time in a functional cell. You have um, 23 pairs of chromosomes, and they're all just kind of in this cloud of DNA called chromatin. And so we need to make what's called a messenger RNA copy, and then that messenger RNA copy, so we'll make a copy of a certain sequence, certain part of the DNA, so that might be a gene right there, and we're going to make a protein. So there's the code for whatever the protein it is we want to make. So we make a messenger RNA copy. That messenger RNA moves out, and then it binds to a ribosome. Okay, so the ribosome can be either free floating. Okay, so if we have here's our ribosome, okay, and our messenger RNA, I'll make it red. A ribosome will find it. Okay, so this is our messenger RNA. So that's the copy of the gene that we just made from there. I made it a little bigger. And then we're going to, from that, build a protein. And these are amino acids that are all going to come together. So it's reading this messenger RNA to make a protein. And remember, what's a protein? A protein is a chain of amino acids. Okay, so that's all that's going on here. So it's being translated. That's the word we use for that. We say that it's being translated. All right. So this can happen. So sometimes we need enzymes, we need proteins that just float around in the cell. So glycolysis, the anaerobic way that we can make ATP. We make ATP without oxygen sometimes. So glycolysis needs proteins, and I'll try to draw them in here, but just sort of floating around. So sometimes we need that. And so the ribosomes will just be floating around too. And so that's where we get into cytosol, the fluid in the cell. This is where soluble proteins are made. So we just make those out in the cell now, or out in the cytosol. Now, sometimes we need, and this shows it nicely here, we need proteins that are going to be excreted, okay, or secreted. So in that case, we need them. If we work backwards, if we see these secreted proteins, and this could be the insulin that I was talking about earlier. At one point, it was in vesicles, okay? So it was hidden in vesicles, and we say, well, how did it get into the vesicles? Well, if you go backwards, sorry to do that to you, this is the Golgi apparatus, and from the Golgi apparatus, or before it gets to the Golgi apparatus, it was in the endoplasmic reticulum. So that means if we have something that is secreted, then it can't just be made out here in the cytosol, it actually has to be made and put into the endoplasmic reticulum. Okay, so do you see that? So that means the ribosome itself has to stick to it, and that's where we get into rough endoplasmic reticulum. And you can see these ribosomes, there are some right there, and that's why we call it rough, because it's got these ribosomes sticking to it. But these ribosomes are just injecting, okay, so here's our ribosome that sits there. It's just injecting the protein that's being made directly into the ER. And then the ER can form these little vesicles and bleb off and then join with the Golgi, and then from the Golgi they can make another vesicle that then fuses with the membrane, and boom, you have the release. Okay, so the rough ER is where membrane bound and secreted proteins have to be made. So if it's not soluble and it has to be bound to a membrane or it's going to be secreted, then you have to get the endoplasmic reticulum involved. Okay, now we do have something called smooth ER, and remember when a cell starts out this big and it's got its phospholipids that are surrounding it, and then it wants to get bigger and bigger, and so it's growing. Well, that means that somewhere from somehow we had to increase the number of lipids. Okay, we had to add lipids to this outside membrane. Well, that's what smoothie R does. Okay, it has the enzymes to produce fatty acids, attach them to the phosphate head group, and then make them part of this membrane. Okay, and then that membrane will move to the outside, and then we'll fuse with it, and then it gets bigger. Okay, oops. I was supposed to just erase a little part of that, but I erased, and there it goes again. Okay, bigger. Okay, so that's what the smoothie R is doing. The Golgi apparatus, now I mentioned that, where proteins go to be excreted. Okay, or they can be moved to the cell membrane. So you can either make them part of the membrane. So if we're looking down here, we can either make them part of the membrane, then they're part of this vesicle, and then when they come over here, then they're still part of the outer membrane. Or we can squirt them into, I'll use this down here, squirt them into the vesicle, into the Golgi. So they were in here, in the Golgi, and then they're in the vesicle, and since they're in the vesicle, when the vesicle fuses, they're released. Okay? All right, so the cell needs protein so it can do its job, harvest energy, and to make more protein. So all of these functions, all of these things we've just been talking about, if you can wrap your head around that, have required protein enzymes and things like that to make the proteins. So you need proteins to make proteins. It all starts out with that little egg and that little sperm and that first little blastocyst, that first little cell that it makes. It already has a bunch of proteins turned on that can allow it to keep dividing and dividing and dividing, so it can make more and more proteins. Okay, and then we'll talk about differentiation later. All right, so proteins are diverse in their functions. For some reason, I only have membrane-bound proteins on here, but there are also proteins that are floating around in the cytosol. Here's a glycoprotein. Remember, that's the one that has the sugar on it. Here are some attachment proteins. Integrins and cadherins, but these are showing integrins, which are just attaching this cell to something in the outside, in the extracellular space, and so it's kind of anchoring it, and it's keeping it there, and it might be joined on the inside to part of the cytoskeleton, on the outside to this, and that's a cell that's not going to go anywhere. It's stuck in that certain place. So attachment proteins, channels, we have pumps, okay, and over here is an example of the sodium-potassium pump, or the sodium-potassium ATPase, because it burns, or it hydrolyzes a molecule of ATP to pump, remember, where sodium is highest, outside the cell, so it pumps let's see, it looks like, yeah, it looks like this is going to be extracellular. So it's taking 3 sodium and pumping them out, and it's taking 2 potassium and pumping them in. And this is why we have that difference, concentration difference of sodium and potassium. Okay, that's why sodium is higher on the outside and potassium is higher on the inside. And this is a very important process. A huge percent of the calories that you eat every day go into driving the sodium-potassium pump in all of your cells. You have to have that concentration gradient set up with sodium higher on the outside and potassium higher on the inside. You have to have that for the cell to function properly. Okay. All right. So we have to make this ATP. ATP is, I like to sort of introduce, we've probably learned since, I don't know, grade school, that ATP is the battery of the cell, or the battery of the body. I don't know, however you learned it, but there's a reason for that, and it's because ATP is just this little molecule called adenosine, and it has 3 phosphate groups stuck to it. Okay, where we really get our energy are from glucose primarily, but that's a 6 carbon sugar. Okay, and it's got a little a little group out here too. So that's a fairly big molecule, glucose, and it's got a lot of bonds and it's got a lot of energy. If you've ever burned a marshmallow, which is pretty much straight sugar, you can see all of the energy that's contained in these sugars. And and then fats, and fats are, long chains of hydrocarbons, and those are big too. So it takes a lot of steps to actually harvest the energy out of this. We can cut up a fat and we can get energy. But then how do we store it? And we store it in this nice little, and I mean, it's a fairly small little protein, and it takes one step. Okay, so to make to get the energy from glucose, it takes about 20 to 30 steps to do it, and it takes a while. Okay, you can get a lot of energy out of it, but it takes a while. Same with fats; it takes a long time to get that energy out of fats and glucose, and even proteins have some energy in them. Adenosine triphosphate, ATP, one step, energy. That's why we use ATP, because you just need to cut off that end phosphate and boom, out comes energy. Okay, so that's why we use it. So we use it to power protein pumps, enzymes, things that need energy, like a muscle contraction. You need energy for that, and rather than going through 20 or 30 steps to get the energy from glucose or fats, we use a lot of ATP molecules. One step and it's done. Okay, so there are two places that energy is made, or ATP is made. The first one is in the cytosol, and that is anaerobic. This is going to be very, very important. Anaerobic. Okay, so some ATP is made there, and we're going to talk primarily about glucose because that's what goes on in glycolysis. You're breaking down glucose, and you get from glycolysis, you get 2 ATP. Okay, yields 2 ATP, and it's nice because you don't need oxygen. Okay. Now, this is where things can start getting thrown out of whack, okay? Because if you have oxygen, I'll say this real quick, if you have oxygen, you can move down to this step, and that takes place in the mitochondria, and that's cellular respiration. If you have oxygen, okay? So let's say we do have plenty of oxygen, we're breathing just fine. When oxygen is present, this process of breaking down the glucose makes a couple of pyruvate molecules. Those pyruvate molecules move down here to the mitochondria, and that's where you have the Krebs cycle, also known as the citric acid cycle, and you have oxidative phosphorylation, or the electron transport chain that happens down here. And from there, look what you get. If you have oxygen, why is this so important? Because lack of oxygen causes a lot of problems. One of those problems when you are lacking oxygen is, I'm going to erase some of this stuff here. One of those problems is a pH change, because look what gets made when there is insufficient oxygen. The pyruvate doesn't move down to the mitochondria. Instead, it's converted to lactic acid. Guess what that does to your pH? It causes it to plummet. Okay, and it becomes acidic. The inside of the cell will become acidic, and all those poor little proteins that are trying to function, and all of those structural proteins that are trying to hold everything together, now are operating poorly. Okay, they aren't able to do their job the way they're supposed to do their job. The sodium potassium pump may not work properly, and everything is just completely thrown out of whack. So you don't want that to happen. Okay, so but normally when things are functioning properly, the mitochondria this pyruvate moves into the mitochondria, and you make a whole bunch of ATP. You don't have the buildup of acid, and everything is chugging along smoothly. Okay, so again, cells must function properly. We talked a little bit about how salt concentrations, how you need, a certain amount of sodium. You need the sodium outside the cell. You need potassium inside the cell. And chloride and calcium, same thing with calcium. There's some calcium out here, but there's no calcium inside the cell. Shouldn't be. Now, what that means overall, though, is that we have, yes, we have ion concentration gradients, but I also mentioned earlier that even though the identity, so I'm going to try to draw this, even though the identity of the molecules inside and out are different, so we can just draw it showing sodium and potassium, or whatever you want the blue and the green to be. If you look at this overall, and this is important, this is how water looks at it. This is how water looks at your cells. It says, oh, look, the concentration outside and inside is roughly the same. So I am water. So that means I can sort of freely move in or out. There are aquaporins that let me do that. I can move in or out if I want to. Well, since the concentration is the same inside and out, that means roughly as much water that goes in is going to move out, and the cell is going to be just fine. And we say that it is, in this particular case, the cell is in an isotonic environment. The concentration inside and out is the same. Now, if, oh, I erased it, sorry. If, and as I continue to erase it, now, if the cell, let's see, I'm just going to draw it again real quick. If I have too many, if I have more of a concentration inside than I have outside, then I'm going to say the water is going to look at this and it's going to say, oh, look, there's a higher concentration of particles, doesn't care what they are. They can be anything. They can be lots of different things. But it's just going to look overall and it's going to say the concentration on the inside is higher than on the outside. So that means that this cell is floating around in a hypotonic solution. Okay, so it's hypotonic because the concentration is lower out here. All right, so what that means for water is water says, oh, you know, I can go anywhere I want to, and I want the concentration to be the same inside and outside. So here's what I, as water, am going to do. I can go in or I can go out, but I'm going to go in more than I go out. And what's that going to do to the cell? Hopefully you guessed it, it's going to cause that cell to expand. It's going to take in all that water. Now that's good because all of these molecules that were really close together are now able to spread out, and so what you end up with is the concentration being the same inside and out. Now I'm not going to draw all this again, but the other thing could happen if the concentration is higher out here. The concentration is higher on the outside of the cell, or hypertonic. So if we're hypertonic, then the water is going to move out of the cell. And that's what you see here. A hypotonic solution means the concentration, you can see this little guy, the concentration isn't very high outside the cell, so the water, so those particles in there are going to, and this is the way I think about it in my head, are going to work like a sponge, and they're going to soak up that water, and the cell is going to swell. Okay, this is an isotonic solution, concentration same inside and out, it's fine. This one, the concentration on the outside of the cell is higher, so what's going to do is it's going to pull the water out of this cell out here to try to even out that concentration. Okay, so these particles, these non-penetrating particles, almost work like a sponge, and they soak up the water wherever the concentration is higher. Okay, so we call hypertonic, we're referring to the particles. Hyper meaning higher. If it's a hypertonic solution, that means the particles concentrations are higher on the outside. Okay, so again, going back to, I mentioned most of these things, but again, going back to pH, pH and waste removal determine how healthy a cell is. The pH determines, or the function of proteins is ideal at their predetermined pH. Now, I say predetermined because usually when we're talking about blood, we want to be in this range, 7.35 to 7.45. But things like in the stomach, where the pH is about 2, there are enzymes that work ideally. They like a pH of 2. Okay, that's where they function the best. These aren't supposed to be 8s. These are supposed to be proteins, but that's where they're happy. Okay, they're happiest at a pH of 2. For the most part, though, most cells want to be, or most proteins are active in this range because most proteins are in your interstitial fluid or something like that. They're in your body. They're not stuck here in the stomach where the pH is 2. Okay, so that's why we want to keep that range right there. So functions of proteins, channels, enzymes, structural, and other chemical reactions only occur properly at the correct pH. Waste elimination, same thing. If you have byproducts of chemical reactions, must be dealt with. You don't want a bunch of trash and wastes building up, particles, pieces of proteins, uh, peroxide, hydrogen peroxide, anything that's not supposed to be there, you want to deal with that. And that's what these guys are for, the peroxisomes, lysosomes, invading bacteria and viruses have to, a lot of times those are taken up by these and they're destroyed. So you can see here, this is taking something up and then it's mixed with a lysosome and then it's dead. Everything in it is gone. Okay, so that's it for this. There are a couple of questions. True, false. ATP is produced without oxygen. Yeah, sure. Yeah, that can happen, and we call it glycolysis. And where does it happen? It happens in the cytosol. Okay. What if you don't have oxygen though? So normally glycolysis produces pyruvate, but if you don't have oxygen, then it produces lactic acid. Very important. Okay. True, rationale, ATP can be produced in the cytosol without oxygen during glycolysis. Your patient has been given an intravenous solution of water. What will happen to the red blood cells? Okay, so here's a red blood cell, and before you've given it the water, the concentration inside and outside, I don't know if it looks that way, but is the same. You're in an isotonic solution. Well, guess what? When you put in water, you're effectively diluting the outside part of the cell. Okay? If you add water that doesn't have enough dissolved particles in it, all of a sudden now, oops, didn't mean to erase that. All of a sudden now you have a hypotonic solution. If I turn my pen around, it deletes things, and even if I get close, they go away. So now you're in a hypotonic solution, which means that water is going to rush in. Okay, it says, oh, look, the concentration in here is so high, and the concentration out here is so low. I'm water, so I can move wherever I want to, and I'm going to try to fix this, and the cell will swell and possibly burst. Okay, and all of that will just leak out, and you've destroyed a perfectly good red blood cell. Okay, so they will burst or lyse. Okay. True or false, if a cell does not make adaptive changes as a result of stress, it will die. Yeah, that's what we're talking about here. We have to be able to fix this. I mean, the cells get out of homeostasis all the time, but they try to do everything they can to maintain that homeostasis, to get back in those areas. Okay. If the cell makes no changes or makes maladaptive changes as a result of any kind of stress, it won't survive. Okay, that's it for this section.